Hi all, um, my name is Janine Seetahal, and as the title of my presentation suggests, I'll be speaking a bit about the origins of this pandemic and going through some of the more recent theories on how we got here. Coronaviruses. What are coronaviruses? Coronaviruses are a large family of viruses found in many different species of animals. Although they may be associated with asymptomatic or subclinical infections, they generally cause gastrointestinal or respiratory infections, and these can be acute and severe or mild and chronic with prolonged shedding. Some examples of diseases that they cause in livestock include transmissible gastroenteritis and porcine epidemic diarrhea in pigs, and infectious bronchitis in chickens. So they're divided into 4 genera: alpha, beta, delta, and gamma coronaviruses. Current phylogenetic analysis indicates that all coronaviruses currently known to cause disease in humans belong to the alpha or the beta group. And sometimes recombination between these similar but different viruses can create novel strains with the ability to cross species barriers. So quickly, I just wanted to point out that the novel SARS coronavirus has 4 structural proteins, and this includes a spike protein, which I will speak more about later in this presentation. SARS coronavirus 2 is the 7th coronavirus known to infect humans. Of the previous 6 viruses, 4 are endemic within the human population and cause mild respiratory diseases such as the common cold. The other 2, which are SARS and MERS, are associated with significant outbreaks in humans within the last 2 decades, and they have caused serious morbidity and mortality. These viruses, SARS and MERS, were thought to originate in bats. SARS emerged around 2002-2003 and was thought to first infect civet cats before jumping to humans. Whereas with MERS, which emerged around 2012, dromedary camels were thought to play a role in the disease transmission to humans. The newly emerged SARS coronavirus 2 was found to share about 80% similarity to the original SARS virus, and is therefore thought to follow a similar zoonotic pathway to human infection. For a successful zoonotic shift of an animal virus to humans, the virus needs to have the ability to infect humans. Sometimes the animal virus already possesses traits that would allow it to do this because of shared features such as receptors between animal hosts and humans. However, more often the virus needs to undergo genetic changes or mutations to acquire these traits, either prior to entering the human host or during human-to-human transmission. Coronaviruses can gain a genomic fragment from another coronavirus strain when they are co-infecting the same host. And this process is known as recombination, and it is important for viruses to expand their species range or switch ecological niches. So why determine origin? When a new virus is discovered, it is important to understand where it comes from. And this is critical to be able to identify and isolate the source to prevent further introductions of the virus into the human population. It also helps to understand the dynamics of the beginning of the outbreak, which can then be used to inform the public health response. And of course, most importantly, as we can all appreciate now, understanding the origin of the virus may also aid the development of therapeutics and vaccines. Geographically, China is the 3rd largest territory and is also the most populous nation in the world. It experiences diverse climatic conditions, which brings about great biodiversity, including more than 100 species of bats. which may often live in close proximity to humans and livestock. And of course, one of the most unique characteristics of China is the Chinese food culture, in which live slaughter of animals, including many wildlife species, is widely practiced. According to numerous previous publications, one of which is exampled here, these factors have flagged China as a hotspot for disease spillover and outbreaks. And given the previous disease outbreak history within the last few decades, it was generally believed that bat-borne coronaviruses would re-emerge here to cause the next disease outbreak. And in this regard, these predictions have in fact been correct. To identify the source of a virus, it is helpful to look at the genetic makeup to see whether it resembles other known viruses or not, which may provide some clues as to its origin. Viruses that are genetically closely linked tend to come from a similar source or similar geographic area. When the genome of this new virus was analyzed, it was found to be related to the original SARS virus, as we mentioned before, with about 80% sequence homology. So it was therefore placed within the same viral group and named SARS-CoV-2. But it was most closely related actually to a SARS-like bat coronavirus named RATG13, which was isolated from a Rhinolophus species bats from Yunnan, in 2013, sharing about 96% of their whole genome identity, with one major difference. This major difference was in the RBD, or the receptor binding domain region of the genome. So, if you would recall, we have these spike proteins on the outside of the coronavirus, and these bind to the host receptors. So we can see the spikes here, and they bind to the host receptors. The host receptors are called ACE receptors. These receptors mediate the entry of the virus into the host cell, and the RBD region, or the receptor binding domain region of the spike protein, facilitates this binding. So you can see the receptor binding domain of the virus here, and this interacts with the ACE receptor of the host to facilitate entry of the virus. Genetic analysis of SARS-CoV-2 found that the spike protein appears to be optimized for binding to the human ACE2 receptors, with mutations in the RBD, or the receptor binding domain of the virus, with 5 out of 6 critical amino acids being different from the original SARS virus, as well as a cleavage site at the junction of the spike protein subunits. So that's between the subunits here. This is the spike protein. These are thought to have positive effects on viral infectivity and play a role in host range. So, what does this mean? Well, for one thing, while speculations, rumors, and conspiracy theories circulate that SARS-CoV-2 is a virus that was developed in a lab, the scientific data points to a lack of credible evidence to support this claim, and is based on this genetic analysis, which indicates that the optimization of the viral spike protein for efficient entry into the host cell is most likely the result of natural selection, and that the virus backbone is totally new and has never before been documented, which would not be the case if genetic manipulation had been performed, as one of the existing beta coronavirus, viral templates would have had to been used. And it also means, given the similarity of SARS-CoV-2 to previously identified bat coronaviruses, and given the knowledge of previous coronavirus outbreaks, we can appreciate that bats are the likely reservoir for this virus. However, the RBD portion in the bat genome is not optimized for efficient entry into human cells, as it is in the novel human coronavirus. So as with SARS and MERS, an intermediate host which provides this genetic material and therefore will facilitate the transfer to humans was proposed. The Malayan pangolin was found to harbor two groups of coronaviruses, one of which exhibited strong similarity to SARS coronavirus in the RBD region, and therefore they have been suspected to be an intermediate or an amplification host which facilitates assortment in this RBD region. However, further surveillance is of course needed to understand their role in the present situation and risk of future transmission to humans. Bats have been proposed to play a vital role in coronavirus ecology and evolution, and may be the original host for many, if not all, alpha and beta coronavirus. Along these lines, another study, while acknowledging that an intermediate host still cannot be ruled out, indicated that SARS-CoV-2 independently evolved in the horseshoe bat before a direct spillover into humans, and that bat coronavirus RATG13 was a sister lineage of this virus, not an ancestor as previously taught. They suggested that high levels of coronavirus co-infection with a large viral pool in the bat can generate novel allele combinations and substantial genetic diversity on its own without the need for interspecies recombinations. So let's just summarize. We've seen that there are few possibilities for how the virus originated and moved from wildlife to humans and onwards to sustained human-to-human transfer. One of the things that stands out for sure is the extraction of wild animals from their natural habitat and mixing of species in common urban settings, such as wet markets and such as that happens with the wildlife trade, which facilitates viral species jumps from animal to animal and from animals to humans, is important in this whole scenario. However, we still have not yet found the smoking gun. Scientists have yet to isolate an intact virus in pangolins or bats or another wildlife species that contains all the pieces of the SARS coronavirus 2 sequence. But as more viruses are sequenced and data are collected, we can piece together a more accurate story of the emergence of this virus. Nevertheless, regardless of the origin, neither bats nor any other animals currently play any role in the progress of the pandemic, which is at present based exclusively on person-to-person transmission, and human behavior dictates this rate. Bats provide enormous ecological benefits. Bats carrying coronaviruses in the wild, undisturbed by people, are not a threat to human health. But we've been here before, and this is not a novel event, as we know. It is the result of human choices and activities For example, human encroachment into biodiverse areas increases the risk of spillover of novel infectious diseases, and this is done by enabling new contacts between humans and wildlife. So unless something changes, and soon, this will happen again. And so maybe the story is to be continued. Until next time. Thanks.